So, given what we just talked about in the last videos, we know a fair bit about the pathogens that can cause disease. So how do we prevent disease? So when we think about all those methods of transmission that we can, how easy it is for us to pass disease from one person to the next, what can we do to prevent it? Well, we've done a lot of things. In terms of public health, we've taken drastic steps in the last 50 or 60 years in order to ensure that we get a healthier life to live. And we started with stuff as simple as properly disposing of feces, sewage, and garbage, so that we don't attract pests. We limit the contact we have with potential vectors like mice and rodents and rats and insects, and that those just sanitary conditions that make life a bit healthier, easier to live. We look after our water sources, purification of water, so our houses have clean water that we can use to drink and bathe. We prevent food contamination, so we have whole branches of government that go around and say whether or not a restaurant or a supermarket is safe to serve food. A lot of the students that I've taught in the past have gone on to do food inspector certificates, and they go around and they make sure that Qatar has healthy, clean food to eat. We have pasteurization of milk, which is a process that takes away pathogens from milk. It makes it safer for us to drink. We have immunization programs where we take vaccines to prevent spread of diseases. And then we have other methods that stem around control of infection that we can do personally. Hand washing, for example, has never been more important than it is right now to prevent the spread of COVID-19 or SARS-CoV-2. And then there's a lot of occupational health and safety laws and rules that go hand in hand with anywhere you go, really, in order to make you safer and healthier. These will also prevent the contamination or the passing of disease from person to person. So why is disease so well spread despite all of those methods? Well, the thing about it is these pathogens are quite good at what they do, and what they do is they find people that they can affect. Some of these reasons go in their favor. For example, the world's population has never been higher than it is right now. And in places where people are really concentrated, that's where disease thrives. It's really easy for disease to spread if there's a lot of potential victims for that disease or that pathogen to infect. We have increased contact with animals in a lot of cases. The example given here was HIV was originally found in chimpanzees, and then it transferred from chimpanzees to humans. Of course, COVID-19 was first isolated from a wet market in China, so we suspect it was from a bat or maybe a pangolin that was eaten from this market in China, and it mutated and it caused an infection in humans. And from there, we know what we're in for today. We increase in travel. It's so much easier for people to travel the entire world right now. If people can travel the entire world, diseases can travel with people to the entire world. The example here is SARS-1, and this was back in 1990, I think. SARS-1 hit Canada, so there was a case in Canada only days after the isolation in China. And if you look at the geographical distance, China is a long way away from Canada, but it made its way to Canada on an international flight. If we look at SARS-CoV-2, the virus that causes COVID, this time last year, nobody had ever heard of it. It never really, it didn't exist. Maybe it existed, but not in the form it is right now. And then by December, we had cases of pneumonia in China. By January, it had spread to a few countries. And by March, almost the whole country had shut down. I know the schools in Qatar had. And right now, from a virus that didn't exist this time last year, we have 33.2 million confirmed cases, and we have over 1 million people dead. So these viruses can travel really, really fast if they're given the opportunity. Medical advances will keep people alive a lot longer. We're lucky that we live in a world of advanced medicine. We changed the way we handle food. Before, in your grandparents' time, they likely ate food that was grown in or very close to the place where they lived. Now we eat food from all over the world. And on top of that, we have these emerging resistant organisms. So if we think about some bacteria, if we get a bacterial infection, we take an antibiotic and we kill those bacteria. But now we get bacteria that that can't be killed with the antibiotics we have. They're too strong. They're too powerful. One example there is MRSA, which of course is methicillin-resistant Staphylococcus aureus. It's one bacteria that we don't really have effective drugs to treat. So how can we control microbes? And if we think about this from an organism point of view, then there's three things that we can look at. First, we can look at sterilization. Now, sterilization is a process where we remove every living thing from an object. So that object would be completely clean of any microorganism whatsoever. This is difficult to do. We can't do it with us, for example. We could never do that. We often use a device called an autoclave that does that. It works at very high pressure and very high temperature. But even sterilization in an autoclave might not be able to kill prions. So we need to be cognizant of that. We have a process called disinfection. Now this is where we use an antimicrobial agent, probably a chemical, to try and kill pathogenic organisms that we find on inanimate objects, so things that aren't living. So if you think about what you do in the lab every day, when you wipe down your bench with an antibiotic wipe, that's disinfecting. Okay? An antiseptic is the same idea. We're going to remove microbes or prevent them from at least reproducing, but we do this on our living tissue. So when you add the alcohol hand sanitizer to your hands, that's a form of antiseptic. So sterilization, disinfection, and antiseptics. Those are three ways that we can hopefully control microbes. Another way of antiseptic, of course, is soap and water, and that's probably one of the most important things that the WHOs have been impressing on us about treating COVID. Now, how do we fight microbes? Well, from a disease perspective, we try and use drugs first and foremost, and we have some drugs that deal with different categories of pathogens. The most common one, and probably what was called the super drug 80 or 90 years ago, was the antibiotic. So this is a really broad group of compounds that target prokaryotic cells in bacteria. So if you take an antibiotic, you need to take it for a bacterial infection. An antibiotic is no good against a virus because a virus doesn't have the same targets that a bacteria would. So the examples of antibiotics would be penicillin or vancomycin. There's these septa drugs, and there's a lot of different categories of antibiotics, although we're running out. We don't have an infinite supply of different ones, that's for sure. The next group of drugs we can have are antivirals. Now these aren't quite as successful because a virus is a very difficult thing to kill because it's not alive. This is a small number of compounds, and they function to target and specifically block the metabolism of a virus in different ways. So it can block the removal of the protein coat of the virus so that it has trouble entering a cell. It can damage the nucleic acids so that it finds it very difficult to take over that cell, blocking enzymes so that it can't produce new copies of itself. So these are ways that we can target a virus. But antivirals drugs are still not as great as they could be, or we hope them to be. And then we get two more terms down here that are similar but just a little tiny bit different. If you look at the front of the words, the bacteria, that's going to refer to bacteria. And then the back part of the words are going to mean two different things. Cidal means to kill. And you may have seen side if you think about suicide, which of course is killing yourself. But a bactericidal agent is one that's going to kill bacteria. Okay? A bacteriostatic agent, now static means to stop or slow down. A bacteriostatic agent is going to kill some bacteria, but mostly prevent them from growing and from metabolizing. So a bacteriostatic agent is not going to remove all the bacteria from an object, but a bactericidal agent would. So these are used to describe different chemicals and different drugs. If it's bactericidal, it'll kill bacteria. If it's bacteriostatic, it'll slow it down and prevent its growth and metabolism. Now, one of the big things about people who work in hospitals as technicians is that they're in charge of identifying these pathogens. So somebody comes in sick, we need to find out what's causing this disease so we can treat it. So the first thing we'll do is we'll obtain and we'll label specimens from patients. Those specimens, those potential pathogens, they'll be taken out to the lab and they'll be grown. So we'll put them in a media, lots of food and right temperature and moisture so that these pathogens can grow. And when they do, we'll isolate them. Very often, if we take a sample from somebody, there's going to be more than one species there. Now, we know that some of these species are good for us, so we don't want to damage them. But what we'll do is we'll isolate the individual colonies, and then we'll multiply them so that we can get growth of individual species that were there. We can stain those cells, like the gram stain, like we've seen in the lab earlier, and that'll give us some information about what those cells are. And then we can do tests to identify an organism. Now, identifying a bacteria is not something that's really easily done in lab. It takes a fair bit of skill. It takes a lot of advanced techniques. It takes a lot of different types of equipment. So it's not something we would get into in this course. But there are technicians who are trained for this and who are very good at it. And their job is to do all of these things so that they can identify a pathogen really quickly so that doctors can then treat it. So even though we're not going to be identifying these microorganisms later in your program, you will get a chance to isolate these bacteria on different types of media. You'll get a chance to do some staining techniques. We'll understand the physical characteristics, so describing the color, the shape, the arrangement. You'll get some antigen-specific reaction tests, and there's a lot more things that you'll do, although typically identifying a whole bacterial species is something that's a bit harder for us to do in an undergraduate situation. Let's skip the slide. So some of these lab investigations, this is one that I've actually done. A student brought us in a bacteria. I grew it. It changed the agar to a weird color that I hadn't seen before. So I grew it. I isolated it and then I stained it. So if you look right here, these little tiny cells are bacilli. So I know I've got a gram-negative bacilli, but I still didn't get it identified any more than that. So these are the type of things that you would do in a lab in order to figure out what organism you're working with. This is another plate of bacteria that we just sampled from the environment. And if you look on this, it's January 16th. MK, so I did this sample and it looks like I took the sample from my beard. So like I said earlier in these videos, bacteria grow everywhere, and this is just an example of where we can collect bacteria. And then of course if we wanted to, we could grow these out and then we could stain them to figure out what they were and possibly identify them, although I suspect they're going to be just harmless normal flora that occur on my skin. And then finally, these are some isolated colonies of bacteria. This is a technique called a streak plate, and if you do microbiology, you'll get a chance to do that. And the idea behind a streak plate is to get these individual colonies, and then hopefully, if you don't see any differences between these, it's safe to say that it's probably one species. So we collect that, and then we try and work with what we call a pure colony. But these are all techniques that we use in a lab in order to identify a pathogen, and then once we identify a pathogen, then we can move to treatment.